Hello and welcome to session 8 of hello and welcome to session 8. Uh, the topic of today's discussion is citric acid production, industrial production of citric acid. So citric acid is the most important commercial product which is found in almost all plants and animal tissues. The molecular formula is C6H8O7. It is 2-hydroxy 1,2,3 propane tricarboxylic acid. It is widely used as organic acid in the field of food and pharmaceuticals. In 1784, Scheele for the first time isolated from lemon juices as calcium citrate, which treated with sulfuric acid gave citric acid in the liquid phase. Citric acid is acidulant in food, confectionery, and beverage. It is also used in pharmaceutical. It is used in industry to complex with metals such as iron, copper to be stabilizer of oils and fats. The microbes used for the citric acid production are, so we have yeast, we have bacteria, and we have fungi. So the yeast are Candida tropicalis, Candida oleophilia, Candida guillermondi, Candida citroformans, Hansinula anamola, Yerovia lipolytica. So, and the bacteria are Bacillus licheniformis, Arthrobacter, Paraphenans, Cornybacterium species. So, all these are the bacteria that are widely used for the production of citric acid, and fungi, Aspergillus niger. So, the majority for the production of citric acid, Aspergillus niger is used, or Candida species is used, and in bacteria, Bacillus licheniformis is used, or sometimes even Cornybacterium species are also used. The organism, Aspergillus, it looks like this. So this is a spore bearing fungi, and this is Candida yeast. And citric acid production is mixed growth associated, mainly take place under nitrogen and phosphate limitation after growth has ceased. So this is the schematic representation of the main metabolic reactions involved in the production of citric acid by Aspergillus niger. So it starts from any kind of complex carbohydrate. It will be broken down or hydrolyzed into glucose. From glucose, we get it enters into glycolysis, and we get pyruvic acid or pyruvate, and this pyruvate, it enters the Krebs cycle, and in the Krebs cycle we get citric acid, hence it is also called as citric acid cycle. So this is the biosynthetic pathway of citric acid, how it is produced in the organisms. Media requirements for high production: carbon sources like molasses or sugar solution. Sodium ferrocyanide is added to reduce iron and manganese. High dissolved oxygen concentration is required. High sugar concentration is required, and pH should be low. It should be acidic, and the temperature should be around 30 degree centigrade. The fermentation process can be batch or fed batch. Aspergillus niger spores per liter, it should be around 5.25 into 10 raised to 6 may be introduced to the fermenter. Aeration is provided to the fermenter by air sparging. Temperature is controlled by cooling coil. Agitation should be around 50 to 100 uh, RPM to avoid shear damage on molds. Fed batch is used to reduce substrate inhibition and prolong the production phase 1 or 2 days after growth cessation. So the yield is around 130 kg per m cube. Yeah, after the process of fermentation is over, so this biomass is separated by filtration. So usually, if this is a vessel, the fungal moss grows on to the top. So these are the fungal mycelia, and the product, which is citric acid, it is released into the media. It is released into the media. So this fungal mass has to be separated. It has to be separated, and this liquid will be taken for downstream processing, and the citric acid will be uh, it will be precipitated, or it will be removed. The biomass is separated by filtration. The liquid is transferred to recovery process. So this is our DSP process. And separation of citric acid from the liquid and precipitation. Calcium hydroxide is added to obtain calcium citrate. The citric acid gets precipitated calcium citrate, uh, tetrahydrate. Wash the precipitate, dissolve it with dilute sulfuric acid. So to dilute sulfuric acid, dilute sulfuric acid, yield citric acid and calcium sulfate precipitate, bleach and crystallization to get anhydrous or monohydrate citric acid. So this is again the uh, downstream processing. So in the downstream processing, so what we have to do, we have to collect the fermentation broth by removing the fungal hyphae or the fungal biomass. Then after filtration, we have to go for the precipitation. In the precipitation, we are adding calcium oxide. So this binds with the citric acid to form a calcium citrate, and then we have to dilute this by adding sulfuric acid, and then we have to add activated carbon, and then purification by ion exchange chromatography, and then crystallization and filtration, and we get the citric acid production, citric acid as a product. This is again another way of representation of citric acid production. So we have to put the substrate, the raw material like the molasses and the inoculum Aspergillus niger into the fermentation broth fermenter, and then we will allow it to get filtration. We'll allow it to we will allow the fermentation process to happen, and after this filtration and the clear fermented liquor, the recovery can be by precipitation, and this is extraction step, and this is by ion exchange chromatography. We have to for the purification. So this is all about the production of citric acid and recovery of the citric acid by using Aspergillus niger. Thank you one and all.